Molecular basis of smooth muscle contraction. Like all muscle tissue, the function of smooth muscle is to contract. The actin and myosin fibers shorten, effectively shrinking the smooth muscle cell. Different systems use smooth muscle, a type of muscle tissue, to exert pressure on organs and vessels so that they can function. Sheets or strands of smooth muscle cells can make up smooth muscle. Actin and myosin fibers that run through these cells are supported by a scaffolding of other proteins. When ATP is released for utilization by the myosin, smooth muscle contracts as a result of the stimulus. In contrast to the on or off contraction of skeletal muscle, the amount of ATP released by smooth muscle is proportional to the intensity of the stimulus. Unlike skeletal or cardiac tissues, smooth muscle does not have distinct striations that are evident on the cells. This is due to the distinct organizational structures between smooth muscle cells and other muscle cells. In smooth muscle, the actin and myosin filaments are layered throughout the cell, giving it a smoother appearance. In smooth muscle, signals from the autonomous nervous system, such as nerve impulses, hormones, and other chemicals generated by specialized organs, regulate contraction instead of the somatic nervous system's voluntary control. Unlike skeletal muscle, which must contract and release fast, smooth muscle is designed to contract consistently. Smooth muscle functions more like a throttle, like in a car, as opposed to calcium trigger that causes a contraction reaction. I'll mention the following locations in smooth muscle again here in a second. Smooth muscle in the circulatory system is essential for maintaining and regulating blood pressure and the passage of oxygen throughout the body. Even though the heart exerts the majority of the pressure, smooth muscle lines every vein and artery. These tiny muscles have the ability to relax and let more blood flow, or they can contract to exert pressure on the system. According to research, the smooth muscles are activated by the presence or absence of oxygen, and when the oxygen level is low, they alter pressure in the veins to deliver enough oxygen. The bulk of the digestive system is lined with smooth muscle for identical reasons. However, the digestive system's cells respond to different stimuli than the circulatory system cells do. Because of its capacity to contract and retain, the smooth muscle serves a variety of purposes throughout the body. In addition to the functions mentioned a second ago, smooth muscle is also in charge of contracting your irises, elevating your fine hairs on your arm, tightening the numerous sphincters throughout your body, and even pushing fluid through organs. Although smooth muscle does not contract or relax as quickly as skeletal or cardiac muscle, it is far more effective at supplying constant elastic tension. So what's the difference molecularly between the smooth and skeletal muscle contraction? Well, the primary distinction between skeletal and smooth muscle contraction is how calcium binds to troponin during skeletal muscle contraction, and calcium binds to comedulin during smooth muscle contraction. Additionally, during the contraction of skeletal muscle, the release of calcium is triggered by action potentials produced by neurotransmitters, whereas during the contraction of smooth muscle, the release of calcium is triggered by smooth spontaneous action potentials and action potentials produced by the binding of neurotransmitters in the receptors on the cell membrane. The source of calcium in skeletal muscle is derived from the sarcoplasmic reticulum as intracellular calcium, while the smooth muscle has both intracellular calcium through the sarcoplasmic reticulum and extracellular calcium that enters through these smooth muscle cells through calcium channels. The release of calcium in the skeletal muscle is initiated by the action potentials generated by neurotransmitters. Meanwhile, the release of calcium in smooth muscle is initiated by both spontaneous action potential and the action potentials generated by the binding of neurotransmitters in the receptors on the cell membrane. Skeletal muscle contraction occurs through the binding of calcium to troponin, and the contraction of smooth muscle occurs through the binding of calcium to comedulin. By receiving calcium ions, smooth muscle cells produce their own action potentials. This extracellular calcium is thus the second source of calcium ions in the cytoplasm. Troponin and tropomyosin do not exist in smooth muscle cells. The myosin light chain complex, an enzyme that phosphorylates the light chain in the myosin head, is activated by the increased calcium calmodulin complex levels. The phosphorylated head then binds to the thin filaments to start the contraction by increasing the affinity of the active sites in the filaments. A few other differences between the skeletal muscles and smooth muscles is that the skeletal are attached to muscle and bones, but the smooth muscle lines the stomach, bladder, uterus, walls of blood vessels, and more. The somatic nervous system controls the contraction of striated muscles called skeletal muscles. As a result, the synaptic input of motor neurons that innervate the skeletal muscles is what causes them to contract. Those skeletal muscles initiate a voluntary muscle contraction, initiating external movement of our body parts, such as throwing a football. Meanwhile, smooth muscle dictates involuntary muscle contraction, such as internal movement, which occurs in organs. These are things that you don't have to think about, such as digesting food or uterine contractions during ladies' menstrual cycle. The central nervous system is in charge of bladder control. The two distinct divisions of the autonomic nervous system are in charge of controlling the smooth muscles, including the destrusor muscle and the internal urethral sphincter. The person urinates as a result of the parasympathetic nervous system stimulating the destrusor muscle to contract. The internal urethral sphincter is stimulated to contract by the sympathetic nervous system, causing the person to hold their urine. The skeletal muscle is known as the external urethral sphincter is stimulated by the contract by somatic efferent neurons. The smooth muscles and skeletal muscles of the bladder both work to urinate. So, some background information to a study I'll discuss here in a second. The inadvertent loss of urine is known as urinary incontinence. When you cough, laugh, sneeze, run, or do heavy lifting, the strain or stress on your bladder causes you to leak urine. This condition is known as stress incontinence. Stress-related incontinence is unrelated to emotional stress. As the bladder fills with urine, it grows larger. Normally, as the bladder swells, muscles that resemble valves in the urethra, a small tube that transports urine out of your body, remain closed to stop urine leakage until you can use the restroom. Sometimes sneezing, leaning over, lifting, or laughing vigorously, for example, can all increase strain on your pelvic and abdominal muscles when those muscles are weak, which can lead to urine leakage. This sounds terrible, but luckily there are some exercises for the pelvic floor that can help tone the muscles that surround your bladder, your lower body, and vagina or penis, and this can turn remedy the urinary incontinence. More to that on the next slide. So, training the pelvic floor muscles is advised as a preventative and therapeutic treatment for lower urinary tract problems in persons of all ages. Stress, urinary incontinence, urine and fecal incontinence, and sexual dysfunction and prolapse of the pelvic organs are all pelvic floor dysfunctions that can be treated by implementing effective pelvic floor muscle training. Both mentally and physically, people experience the signs of urine incontinence. Uncontrolled urine leakage lowers quality of life and is associated with social isolation, emotional difficulties, and decline in work productivity. Pelvic floor training refers to programmed, repetitive, voluntary contractions of the pelvic floor muscles, following a protocol that determines the frequency, intensity, and type of exercise, as well as duration of training. The proposed training programs typically include one or more sets of exercises per day, performed at least several days per week. In a study I'm mentioning here, it shows the importance of correlation between the smooth and skeletal muscles involved on the pelvic floor. The study included 42 female students from the Gdańsk University of Physical Education and Sport in Poland. Initially, the participants between the ages of 19 and 28 were randomly assigned to the intervention or control groups in a 1-to-1 ratio. There was an incontinence impact questionnaire to assess the quality of life associated with urine incontinence both before and after the experiment. A score between 50 and 70 indicated a moderate quality of life. A score between 50 and 50 suggested a good quality of life. and a score below 50 was classified as poor quality of life. Using a self-applied vaginal probe on surface electrodes attached to the gluteus maximus, rectus abdominis, and external oblique muscles, the scientists evaluated the pelvic floor muscles contraction technique using surface electric myography before and after the experiment's 6-week period. To assess the pelvic floor muscle contraction technique, they used MyoResearch software. Based on a preset threshold for electromyographic activity, the software determines if the selected muscles have been stimulated, and if so, in what order. And this is also referred to as a firing order. The firing order for the pelvic floor muscles, rectus abdominis, external oblique, and gluteus maximus muscles were all taken into consideration while evaluating the technique of the pelvic floor muscular contraction. In order to develop different motor skills, the study employed a pelvic floor muscle training regimen based on graduated strength training. Each week was different, as were the workouts and the amount of sessions. The exercise was intended to release muscles' strength, speed, endurance, while also intentionally bringing them back to relaxation. I won't go into those exercises in great detail due to the presentation's time restrictions. The results are on the following slide here. Okay, so I know this is a little bit hard to see, but I wanted to show the difference between the 3 measured exercises. The quick flicks right here, the first 10-second contractions, which are the right here, and then the 5th 10-second reactions right here. Contractions, sorry. The controlled group's exercises look a little bit more all over the place. So these are the controlled groups right here and right here. These are just kind of like way all over the place right over here, okay? Ironically, they're not very controlled muscle group when doing these exercises. So they kind of just look all over the place. With those that experience intervention, there's more of an exact muscle contraction that makes the chart look more clean and less all over the place. So you see how these look just more so in order right here? These are the ones that went into the therapy. You can tell there that the muscle contraction is very exact and learned. In this muscle group shown on the chart, the changes in the firing order and the technique score of pelvic floor muscle contraction in all other motor tasks except the technique score and the 4th 10-second contraction also had beneficial tendency, but were not statistically significant. In the control group, there wasn't substantial changes of any of the analyzed variables. This chart shows individual changes in the firing order of the pelvic floor muscles in the intervention and control group. A positive result is the reduction in the individual value, which translates into earlier activation of pelvic floor muscles and results in a better technique of said performed exercises. The first and 5th 10-second contractions showed a statistically significant improvement of the intervention group. In the initial 10-second pelvic floor muscle contraction during the post-interventional assessment, 33% more members of the control group completed the exercise using the proper form. In the post-intervention assessment, 50% more women executed the contraction technically sound in the 5th 10-second contraction. These findings indicate a noteworthy advancement in the execution of the activities as a whole. Now, why does this matter? The research confirms that introducing the pelvic floor muscle enhancement exercises into high-impact training reduces urinary incontinence symptoms in young physically active women. The incorporation of pelvic floor muscle training into high-intensity activities prevents the deterioration of pelvic floor muscle function and maintains a good quality of life in relation to urinary incontinence. The pelvic floor muscle contraction techniques was improved by the intervention that was offered, and the changes that were seen were statistically significant. Pelvic floor muscle training added to high-low impact exercise regimens enables physically active nulliparous women to take part in a variety of sports and leisure activities without generating unfavorable alterations in pelvic floor function. Asymptomatic people should be educated about pelvic floor muscles trainings and given guidance on how to use it. Women should be encouraged to attend electromyographic biofeedback sessions and learn how to correctly contract pelvic floor muscles. And these are the resources that I use.